on the Moodle, on the LSEC, and a lot of places. So just send me your email, and then I'll put you in. Yes. All right. So we finished last time. We discussed last time atomic bonds and molecular interactions and chemical building blocks of the cell, and said and introduced chemical equations and chemical equilibrium. Let's say again just a few words about biochemical energetics before we go on to the next chapter about protein structure and function. And by the way, the question page about this chapter, biochemistry of the cell, you'll answer it at home, and we'll answer it together in class next week, not today. So biochemical energetics. So which energies would you expect to find in the cell? Which types of energies? Chemical energy, indeed. Electric. Right, electric. Kinetic, I think it's kind of a mechanical, same domain of energies, I think, should be termed. I don't know, but yeah, but it's also true. So indeed, several different kinds of energies exist. Thermal energy and mechanical, which I think kinetic should be inserted here, I think. Electric energy, chemical potential energy, and concentration gradient. And of course, cells have evolved several machineries that enable them to transform one type of energy to another. Maybe the most, maybe an interesting, and the first example that comes to mind is photosynthesis that happens in plants, where light energy transforms into chemical energy. We'll see lots of examples, neuronal examples later on, but this is generally speaking. Okay. All systems change in such a way that free energy, we're talking here about the Gibbs free energy, is minimized. And and the free energy, the delta free energy of a chemical equation is the free energy of the products minus that of the reactants. So let's just do reactants and the error and products. Okay, and it can also be calculated as written here. So the Gibbs free energy, the delta in Gibbs free energy for the chemical equation is the bond energy, the delta in bond energy, which is also called the enthalpy, minus temperature in degrees Kelvin multiplied by the entropy, by S. Okay, so it's the delta G here is in direct relation to the delta H, to the enthalpy. And an exothermic reaction is a reaction in which the products have less bond energy than the reactants. So if the energy between the two sides of the chemical equation is like this, so this is an exothermic reaction. This will be the energy. Exothermic meaning that it secretes energy. The system secretes energy to the outside. And if it's the other way around, then it is an endothermic reaction. It absorbs energy. If it's like this, for instance, it'll have to absorb energy in order to occur. So this is, again, talking about the enthalpy. And going back to the Gibbs free energy, so it's in direct relation to the delta H, but it's in inverse, because of the minus here, in inverse relation to the delta S, to the delta in the entropy. So the more disorder the system, the bigger this part, and the more minimized we are in the delta G, and so the biological reaction will tend to occur more. And how much should we take into consideration the delta T? That's what the T here gives us. It's sort of a gain button. So when the degrees are very small, when we approach 0, so this factor, this part of the equation almost drops. And delta G is, for all practical means, equals delta H. And in higher than 0 temperatures, which is always the case, we have to take into consideration the delta S. So this is sort of the weight for the delta S. And again, let's give the example we gave last time for a polypeptide that's built of 10 amino acids. So if they are scattered, unlinked before the polypeptide is synthesized, so the entropy is high because each amino acid have high degree of freedom and are free to go around in space. And the enthalpy decreases when the protein is synthesized, and the amino acids movements are restrained a lot.
What is the relationship between exothermic and endothermic and the Gibbs free energy? If it's like positive, then it's always
Right. So exothermic and endothermic reaction will have the same effect, will have the same effect on delta G as they have on delta H, meaning that because of the direct relation, meaning that when you have an exothermic, let's plot it right. An exothermic reaction will tend to have a more minimized delta G. An exothermic reaction, and so will be more favorable thermodynamically than an endothermic reaction. And what happens mostly in cells is that these two sorts of reactions, endothermic and exothermic, are coupled together. So the one gives energy to the other. And we'll see examples later on. Okay, while discussing just a bit about biochemical energetics, we will have to introduce the adenosine 3-phosphate molecule, or ATP, which is the most important molecule for capturing, transiently storing, and subsequently transferring energy. It's termed the energy coin of the cell. And that's the molecule. It has 3 phosphates. And the adenosine-based we already described last time. And between the phosphate groups, we have 2 covalent bonds that are enriched with energy, and can be break down and then the phosphate groups can link to another protein, and changing its energetical level and enable it to be active or to change its activity. So the most common mechanism of energy coupling is phosphorylation, again, which I just described, which is the transfer of one of the phosphate groups to another protein. So let's look at this process, the principal schematic representation of it right here in these chemical equations. So protein B, for instance, together with adenosine 3-phosphate, can be phosphorylated, meaning that one of the phosphates from this molecule, the last one, will be linked covalently to protein B, and we are left with adenosine D-phosphate, or ADP. And then so now the energetical level of B phosphorylated is higher than was before, and it can do interesting things, such as, while encountering C, turn it into D, and we are, and the lone phosphate right here is dissociated from the B. And this reaction was enabled only after this reaction, or thanks to this reaction.
Why does this phosphate give, because this is an exothermic reaction?
Okay.
Or where is the energy coming from?
The energy comes from the breaking of this bond after it is linked to the B.
Okay.
So in order to link this, it's an exothermic reaction, right? And in order to link this phosphate to B, there's another protein usually that's involved in the process and that holds the ATP in the correct orientation and correct distance from the B, from the right site in B, in order for it to be phosphorylated. And then afterwards, it can be broken down and thereby gaining energy to the system, enabling this process, which was not enabled without this B. All right. Okay, so this is like the basic introduction for ATP, and we'll meet this molecule a lot during the course. Okay. So again, please, you can find it on the website, and I'll send it to you by email, the question page for biochemistry of the cell. Also, go over the terms, mainly, of course, those of you who this is the first time that they hear these terms, and you're welcome to approach me with any questions, of course. And that's it. Let's now go on with the protein structure and function, the second chapter of the course. So it's a big chapter. It's parallel to the third chapter in our textbook. And in the end of this chapter, we'll read together an article about the structure and function of the NMDA receptor. So it's a protein, and now we lay the ground to be able to read this article really to the small letters, to understand each and every sentence in it. Okay, So again, it's a big chapter. We'll start with the hierarchical structure of proteins. We will meet the 4 structural levels of proteins. Continue with several modifications, chemical modifications, folding, also regulation and degradation that occurs on protein after they are synthesized. We'll then focus on these 2 protein families, although there are several others in the cell: enzymes that promote chemical equations or chemical reactions, and molecular motors that enable movement in the cell, or what we call cellular motility. Then discuss briefly about common mechanisms for regulating protein function, and then introduce several methods we have in the lab for purify, detect, and characterize proteins. And with that, we'll finish. And again, the climax will be to read together the article about NMDA. Let's start with the hierarchical structure. So the most basic primary structure of a protein is its sequence of amino acids. And then there are a bunch of additional structural levels we will see in details in the next couple of slides, but for now, just let's look at it generally, that give rise to the overall 3-dimensional structure of the protein in space. By the way, the supra, or the 5th, supramolecular, or the 5th structural level that we see here in the slide does not exist in the textbook. It's a terminology, semantic issue. Some textbooks stop at quaternary structure, and others proceed to another 5th structure. But in our case, we will discuss only this one, which include also the features that are presented in the supramolecular structure. Okay, and then the final overall structure of the protein gives rise to its function, or dictates its function. And proteins do a lot of things in the cell. They are responsible for regulation of other molecules, other proteins, and other molecules and other processes. They can have a supportive or structural roles. They're responsible for movement, catalysis of reactions, transport, and signaling between cells. So biologists argue that the function is derived from the 3-dimensional structure. This function is derived from the 3-dimensional structure, which in turn is derived from the primary sequence, the primary structure, the sequence of amino acids. And we say that not because we know how to get from the first level to the last, but because we believe this is true. Okay, sort of. We're still missing information to do it all the way through, but. Okay, so the primary structure is repeated. Before that, let's go back to the definition about the backbone of the protein, which is composed of the amide, carbon, alpha, the central carbon, carbon not central, this sequence of atoms that sit right here, which are the backbone of the protein. One side of the polypeptide will have the amino group exposed, while the other side will have the carboxyl group exposed. So the first end is called the N-terminal for the nitrogen, and the second side, and the other side will be C-terminal for the carbon. And the linear arrangements of amino acids, just the sequence, this is exactly the primary structure of a protein. And the size of proteins or polypeptide is reported as its mass in daltons. A dalton is 1 atomic mass unit. You can read maybe the first paragraph on Wikipedia. Basically, it's defined as the 12th part of carbon in its naturally occurring state. It's something like 10 to the minus of 27 kilograms. So this is, we will use atomic mass unit or daltons, which is really the same for different proteins, or kilodaltons. And proteins can be as long as 4,000 residues, so they can have a very big mass, and they can be long. We'll see it later on.
Residue is just an amino acid?
Right, residue is another word for amino acid here, although we also use residue or as a residual chain when we speak only on this moiety, the green one alone. Okay, so but I'll make sure when I speak about the one and not the other. Okay, secondary structures are various spatial arrangements there are actually local foldings in the protein. And they occur thanks to non-covalent stabilizing interactions. So the primary structure is linked, I mean, the amino acids in the primary structure are linked through covalent bonds. Obviously, here they are, the peptide bond, which was mentioned last time. And now, from the secondary structure and up, these are only non-covalent stabilizing interactions. And when you don't have these stabilizing interactions, which you most usually have, so the polypeptide assumes a random coil structure, meaning that it doesn't look like anything. It's just it doesn't have a defined structure. And what are these non-covalent stabilizing interactions in the secondary structure? These are hydrogen bonds that are formed between certain residues and give rise to well-defined periodic structures that can be one out of three: alpha helix, beta sheet, or short U-turn, U-shaped turn, or just U-turn.
Bonds can only be hydrogen bonds?
Yeah, in the secondary structure case, indeed, yes, that's true. And let's start with the alpha helix. So the hydrogen bond in this structure is the following. It's the hydrogen bond between the carboxyl, the oxygen of the carboxyl group right here, with the hydrogen from the amide group right here. So this is a typical hydrogen bond. The partially positively charged hydrogen is hydrogen bonded with the two unpaired electrons from this oxygen, and you have a hydrogen bond right here. And it usually occurs between two amino acids that are far amino acids from one another. And note that the hydrogen bonds here are between atoms that are part of the polypeptide backbone, and not between atoms that are part of the residual chain. So the outcome is that, okay, so now you have a fragment of a protein or a region in the protein where the polar or the charged moieties that are part of the polypeptide backbone are already engaged in hydrogen bonds between them. So the hydrophobicity of this region will solely be determined by the identity or characteristics of the residual chains. If these will be hydrophobic, then the region will be hydrophobic. If the side chains will be hydrophilic, the region will be hydrophilic. Again, because anything that gives the protein hydrophilic nature is already taken. Okay, so yes, sir.
Are there any constraints on these anodes in the structure? Meaning that some are more likely in this structure and some are less likely in this structure?
Yes, the answer is yes, and we'll see it. I'm not sure we'll see it actually. Yes, so there are maybe in the examples we give. Yeah. Well, it's not only, so first of all, there are some constraints. There are typical residual groups for alpha helix. I don't think I'll introduce it today, but there are. And the second thing is not only the identity of these residual groups is what determines the existence of this alpha helix, but also their position in relation to one another, and also in addition the interactions between this structure and another nearby structure that can stabilize this one. But particularly when you'll see alpha helix, when an alpha helix occurs in protein, so these are the hydrogen bonds that stabilize it. And because the bonds are in parallel orientation or parallel to the axis of the region, if this will be the axis of the region, so it's hard to bend. It's a stable structure all in all, and it opposes bending in this side. It opposes any side force that you'll put on this structure. Okay, the beta sheet have the same hydrogen bonds again between the hydrogen in the amide and the oxygen in the carboxyl. And this time, these hydrogen bonds are between adjacent strands. side view of the beta sheet that's composed of several such strands show that the residual groups will be alternately positioned up and down, upwards and downwards. In a beta sheet, you will usually find something like 3 and more, and there are also 2 strands that exist that form a beta sheet, and between them there is a short 5 to 8 residue polypeptide segment right here. That's also usually is a U-turn, to be introduced in the next slide. There is some nomenclature or terminology about beta sheets. So if I'll say that we're talking about a 4-stranded antiparallel beta sheet, that means that it has 4 strands, and the antiparallel refers to the orientation in relation to one another between these 2 strands. So while the one goes from the C to the N, so the other go the opposite direction from the N to the C, and so on and so forth. So again, hydrogen bonds between atoms from the polypeptide backbone in the beta sheet and in the alpha helix as well. All right. Let's talk about the Ramachandran plot, named after an Indian investigator that invented this beautiful plot. Okay, so in order to plot it, or in order to give the structure of a protein in space, it's enough to talk about about 2 spatial 3-dimensional 2 angles in 3 dimensions, and because it's these are 2, this is an angle not in a plane but in a 3 dimension, so these are dihedral angles, meaning that they are angles between 2 planes. The first one, phi, is the one that involves the backbone atoms Cn, C alpha, C tag. Let's try to use this model, although it's not that big and it's not that stable, but let's try to use it anyway. So what we have here are the residual groups in green, the carboxyl in red, and the rest of the colors I didn't have enough pieces, so it's try to, yeah, okay. So, and anything that's linked to the green, which is the residual group, is the central carbon. So this is C alpha, C alpha, C alpha. And the 2 dihedral angles are the ones that from the left side and the right side, let's not talk about sides, but when I hold it like this, from both sides of the C alpha. So again, phi, the first one, is the angle that is between 2 planes. The plane that's defined by C tag and C alpha, and the second plane, N C alpha C tag. To make it more simple, it's the angle that when you change it, you will rotate around this axis, the N C alpha axis. So if let's take this C alpha and this is the N and C alpha axis. If I'll rotate this angle, this will be changing phi. And when I'll change it, you'll see that I actually determine or control the C to C distance. So now they are farther away, and now they are closer together, these 2 C's. The second one, psi, is the one that's between the N C alpha C tag plane and C alpha C tag N plane, and again, is the one that changes or rotate the C alpha C tag. So again, let's try to make it a little bit linear. C alpha C tag. So when I change this time, when I rotate around this axis, so the 2 nitrogens, as written here, are now closer together or farther apart. All right, so these are the 2 dihedral angles. You can play with it in the break if you want to. And now if we'll take a lot of proteins and each time calculate the phi and the psi, and now plot the one as a function of the other in a scatter plot, we get these distinct islands. And what are these islands? They're actually the angle values around the alpha helix participating amino acids and beta strand participating alpha helix. And actually, if you take this small kacha model and put put psi around 0 and phi around minus 90 degrees, you will gradually build yourself a left-handed alpha helix. And the same thing for the beta strand. And this third distinct island is actually a right-handed alpha helix, which is much less common, but still exists. Because so the psi is relatively the same, and phi is, instead of minus 90 degrees, plus 90 degrees.
And so where do you define the zero of these angles?
When
we are the closest?
Are the farther apart. This is 0. Not this one. I mean, the orientation you see right here. So when they are farther apart, this is the 0. First link right here, so this is 0. So these are when they are farther apart, these two black dots. And now they are close together. This is 180 degrees.
Semi-linear.
Semi-linear.
Okay, and the scatter of dots on the plot is the stable states of the molecule?
Yep, indeed. So it's a stable state folding-wise. You're talking about now the different foldings?
Yeah, geometry.
True, yes. So this...
Stable.
Sorry?
Geometrically stable.
So I'm not sure what you mean here by geometrically stable. So you take a protein with a defined structure, and then you measure the angles between the different atoms in space, and these are the numbers you come up with. So it's a stable, not moving, not changing protein. Yep.
Is this a flow diagram here?
This one, the scatter?
Yeah.
What do you mean by a flow diagram? I'm not sure it is. Flow meaning...
These are all individual molecules.
All right.
Either like the phi or the C.
Right. So each dot here is actually an amino acid for which you calculate or you measured. So you measure the angle psi and the angle phi, and then you come up with two numbers, and you just put these coordinates in space. So each dot here is taken from one amino acid, from one protein. And what I showed from one molecule, one protein alone. But then this scatter, the Ramachandran plot, you can plot the Ramachandran plot for a single protein alone, right? That have here, I would say, 10,000 dots, 10,000 amino acids. But the classical, what we're looking at right here, is a Ramachandran plot that's the average for lots of proteins. Okay, so the first thing to learn from the Ramachandran plot is that a lot of the amino acids, some people give different statistics here, 60, 80, 90% of amino acids are either in an alpha helix or a beta strand. So I'm not sure about the precise percentages, but a lot of the structures in proteins belong to either the beta strand or the alpha helix. And another thing, let's ask what or how would these two amino acids, and now reminder, these are the unique amino acids, how would they look if I now mark in red only the amino acids that are, only the angles that are around either the proline or the glycine. And let's start with the proline. So it's a weird looking amino acid. Why is it weird? Right, because of this last carbon covalently linked to the nitrogen that it's part of the backbone. So that's weird. And again, if I'll just mark in red all the dots that belong to proline, how do you think it will look in this graph? Let's focus on phi, which rotates the N to C alpha link. It's exactly the link we're talking about right here. It's the N to C alpha. That's the C alpha because the residual group is linked to it, and C alpha. So would phi be able to rotate? Right, so it will be relatively localized on this graph, first of all. And nothing we can say it will be more localized around the phi than it will be around the psi. So it will be more localized around the phi, something like maybe a line, a vertical line in this graph. But also because of the phi also, so it affects the psi as well. So it will also be a bit localized in the y-axis. And that's in oppose to the glycine, which will look how in this graph? What do you think? So glycine has just an hydrogen atom as a residual group. Would it be constrained as proline? Sorry. Would it be constrained as proline? No, much less so, actually, and much less than any other amino acid as well, because of the extremely tiny residual group. So it will be scattered here and there in this graph, more than an averaged amino acid. Right. Okay, so that's the Ramachandran plot. Questions? Okay. Turns, or U-shaped turns, the third and last secondary structure. These are composed of 3 to 4 residues, which form a sharp U-sharp sharp bend. And speaking about proline and glycine, they are commonly presented in turns. Glycine is there because it's tiny and it frees the space that needs to compact this region. And proline is there because of its built-in bend that allows stability of this U-turn shape. So that's why each with its own reason is there. And again, we find U-turns a lot of times in beta sheets between the different strands. Just an example of a name for example of a beta sheet. And another more interesting structure that is linked with U-turn shape is the right-hand three-sided beta helix taken from some protein. So it actually goes into and out of the board. These are parallel this time strands, and the U-turns link between strand and then between each adjacent strands, and then you go like a spiral into the board or outside the board. All right. So these were the secondary structures, the 3 common ones. Tertiary structure refers to the overall conformation of a protein in space. And in contrast with the secondary structures, which were stabilized by the atoms from the polypeptide backbone, the tertiary structure is about or involves non-covalent interactions between residual groups, the residual groups per se. If we have hydrophobic interactions, they will be found between if we have stabilized hydrophobic interaction between non-polar side chains, and hydrogen bonds will be found between polar side chains. So again, this time it's interactions between the side chains themselves. And again, in opposed to the secondary structure, the tertiary structure is not that stable, is less stable than the secondary structure, and the stabilizing interactions are weak. And that gives rise to a feature. It's not a bug. It's a feature of proteins that they undergo minute fluctuations all the time. That's important actually, and has important consequences in the function and recognition ability of proteins. So the tertiary structure is, it breathes sort of continuously. Okay, this will be a good time to go over the different visual representations or structural models we see in textbooks, in articles for proteins. The first one is all of them are for a protein called RAS, together with a molecule called guanosine D-phosphate, or GDP, incorporated in it. So the first is the backbone trace model, where you see the coordinates in space of only the backbone of the protein, right? So leaving aside, chopping out any residual groups, and focusing on the backbone itself. The second kind is the ball and stick, which is quite similar to this actually, in which you have the coordinating space of each and every atom, so it's a bit more messy. And ribbons model, which highlights the alpha helices and the beta strands. And the solvent accessible surface model. So this would be how the environment sees the protein, with its bumps and crevices, and in red are regions that are focused with negative charge, and in blue, same thing for positive charge.
And the white thing around it?
Sorry?
Or the white around it?
Is neutral on average.
So like the blue balls that are in all of them is like this chunk of.
These chunks are GDP.
Ah,
What you see here is actually two combined models that you'll be able to visually separate between the RAS and the GDP. So what I just explained was only about the RAS. The GDP is the same thing for all models, just like no sticks, but yeah, a bowl model. All right. Protein motifs. So a motif is a combination of secondary structures, and they are also a signature of a specific function. And so there are a lot of databases exist today for a specific that gives you the function or the putative function of a specific protein motif. And when we see two proteins with similar protein motifs, we will argue or assume we'll try to prove that they have a similar function. Let's see three seminal examples of protein motifs that again are combinations with different numbers and orientation of secondary structures, the ones we've introduced. The first one is the helix loop helix motif. Here it is, the helix loop helix. The amino acids right here in the loop are hydrophilic, and they interact via ion bonds with a calcium ion. And what this structure does, what it gives to a protein, is the ability to bind calcium. And it's important to buffer calcium from the cytosol of a cell, and it's important because it moves, because it turns the protein that this fraction is part of to an on state, to an active state, and other purposes which we'll meet later on. The second protein motif, the zinc finger motif, supposed to look like a finger a little bit, composed of an alpha helix and two beta strands that, and from from the U-turn and the alpha helix, several amino acids protrude and face each other to interact again with via an ion bond with the zinc ion. Proteins that have this usually bind RNA or DNA. And the third example will be the coiled coil motif. It's composed of 2, 3, or 4 alpha helices. Here we only see 2, because we dropped off the 3. 2 alpha helices that in each one you'll find hydrophobic amino acids that face just one side of the helix. So what you get is a hydrophobic line here, or a hydrophobic region that stick to another hydrophobic region right here via the hydrophobic effect. And these two helices now stick together very stably, and that's a very stable structure, which we'll see in structural supportive proteins.
Maybe what the color stands for in each one of them?
So it's the ribbons. It's the ribbons model, in which red is just the alpha helix, and usually blue or something around blue will be for the beta strands. And that's what it stands for here, just to discriminate between the two hydrophobic, the two amino acid sets that belong to the one or not the other. Right, it's a good question. Okay, so, and we call this, or we find this in proteins we refer to as fibrous proteins. In each one, we'll find a consensus sequence. And when we say consensus sequence in biology, we mean that the sequence doesn't have to be identical. Now we talk about sequence of amino acids, but it's the same thing for nucleotides, or nucleotides mostly. So it doesn't mean that it has an identical sequence, but it has a significant similarity between the sequences that are important to give the functionality of this structure. So for instance, in the zinc finger motif, there will always be either an F. These are names of amino acids, right? Either F or Y in the first position, C in the third, 4, 5, 6 position, again C, and so on and so forth. So wherever it's blank, so it's you're free to put whatever amino acid you want to. Or in other words, we were not able to identify any statistically significant identity of amino acid in these positions. Okay, so this was also about the consensus sequences we sometimes refer to in biology. Okay, protein domains are part of the tertiary structure. So, okay, so first of all, the tertiary structure of proteins that are big proteins. This is, relatively a big protein. Is typically subdivided into distinct regions called domains. So it's a relatively loose definition. So don't be, afraid or surprised. It's not. There is no good definition. Just when biologists look at a protein and say, okay, this looks like a sphere or a globe, so let's call it globular domain, and this looks like a cylinder, so this would be the fibrous domain. But so we're now looking at at the tertiary structure level. And we call distinct regions we see with our eyes, we call them domains and give them names. Structurally, here's as close to a definition that we'll get. A domain is a compactly folded region of a polypeptide. Okay, like motifs of secondary structures from the previous slide, domains of tertiary structure from this one are incorporated as modules into different proteins. So when we talk about modularity in biochemistry, we mean that the cell uses building blocks or specific structures in one protein for one purpose and another protein for a similar purpose or even a different one. And we'll argue that during the evolution, when a certain structure came up and was found to be effective and stable and give some function to the cell, so it is used to another protein as well, and to different processes. So, for instance, the EGF, that's the name of a protein which only is built of a single domain, a globular domain. It has a function by itself, and it also can be found incorporated into another protein, a bigger one called neuroligin, together with additional domains. So this is what we mean by modularity at the level of domains and modularity at the level of secondary structures, motifs. And the quaternary structure, so some proteins are not built from a single polypeptide chain, but from two and more. And for them, there is already the quaternary structure that describes the number of polypeptide chains, also called in chemistry stoichiometry. And the relative position of the subunits in relation to one another. Again, the orientation between them. So this is the high level for us of description of a protein structure. Again, this will be very big, unsurprisingly very big proteins in mass and in size. Here is an example, the mRNA transcription initiation machinery. It's not a protein, but rather a protein complex or a machinery that is built of one protein called RNA polymerase. General transcription factors, each of these is a protein, and another bunch of what very general name mediator complex, something that glue the whole thing together. And there are actually also not only proteins, but also RNA molecules embedded in this complex and different kinds of molecules. So explaining the ratio or the number of each subunit, not really subunit, the number of each protein and the orientation in which they link each other, this will be a quaternary structure description. Okay, let's just go over one last slide and say a word about proteins evolution. We can draw evolutionary trees based on the primary structure of a protein. And we can do the same thing based on its structural, three-dimensional arrangement. And actually, we will come up with quite similar trees, evolutionary trees, which give us a credit in our assumption that the second and up structures are dictated by the primary structure. And let's just introduce the term homologs in proteins. So these are proteins that have a common ancestor during the evolution. Now we don't talk about different species, but different molecules. In our case, a protein. Let's see example for the hemoglobin. Yeah, that's a human protein that carries oxygen from the lungs to the peripheral and not only peripheral tissues. It's a tetramer. It's built of 4 subunits. But look how similar one of the subunit is, it has also the alpha, so the beta subunit precisely, to different proteins that exist in different species. Part of them more ancient, the leg hemoglobin and the beta subunit of the hemoglobin and myoglobin. So these proteins would be homologs of each other because we will assume that they have a common ancestor somewhere down or up this line. All right. Let's stop here and take 15 minutes break and then continue with folding and modifications.
סימן שיט עד מתי?
עד רבע ל.
איזה שעה שיעור?
כן, היי.
אני ממש רבע
All right.
How was it the homework assignment?
So it's not really an assignment in this course. So what we have is when we finish each chapter, we have a question sheet, and it's on the web. And then you'll just answer it alone at home, and then we'll answer it in the next class together here.
Okay, so
You see the model, right? So these are these PDFs. Nope.
oh, the question.
Yeah, the question, the Q, right?
Okay, so
The Q files.
so
And
we'll
yeah.
answer them together.
Sorry.
We will answer the questions together.
Yeah, we'll give the answers in class, and so you can answer it at home alone or with a friend, but just make sure that you tried it yourself before we do it together in class.
Okay.
And I'll send this page also by email. Are you in the mailing list?
Yes, I'm in the
Right.
mailing list. Can you explain the angles?
Sure. So let's start with the C alpha. That's linked to the residual group. That's why we know to identify by the way.
Yeah.
The first one, C, is right here. If you hold it, you can go back to definition. Is the one between C alpha and the nitrogen, which I put it as bluish, which is visually the standard. It'll be this. So you need to hold between these two atoms and then rotate. And now you're playing with the C angle.
Okay.
And another way to do it is which sometimes is done, is another plot, not the Ramachandran, that measures the distance between these two atoms, the two carbons.
So this carbon is linked amino group or amine group?
This carbon is linked with the carboxyl. This is here is O.
Okay.
Yeah.
Okay. So this is the carbon from this.
This is the carbon that we call C tag, meaning that it's not central.
Okay. Okay.
So yeah.
Within a chain. So this is one amino acid group.
Yes.
And this is one amino acid group.
Right.
So this is the link. This is the peptide.
Exactly. There's the peptide bond. Maybe I should have put it. Yeah.
Okay.
Right.
And if I rotate this.
Mm-hmm.
Okay. Okay. So between two amino groups, the carboxyl.
It's not between it's not between two amino groups. It's actually inside one amino group.
Mm-hmm.
So we don't talk about an angle between that rotates the peptide.
Okay.
For several reasons. We can do that, by the way. There are three dimensional Ramachandran plots. They're also additions, but it's much less important in the alpha helix and brastrand
So we talk about only this distance. This is okay. This is the one thing in here.
Right. So when you rotate around this axis, it brings these two together
Okay.
or apart.
And then
And then the second angle you'll be playing with if you rotate around this axis, right? So indeed, actually you have three angles you need to talk about, right? In a protein, in a protein backbone, because you have 4 participating carbons. But the polypeptide axis or the polypeptide bond axis is ignored right here in the Ramachandran plot.
okay.
Because it's okay, because it works even when ignored.
Okay. So one this and second one.
And the second one is in the right.
This one.
Yeah, you're right from this one. You rotate this one.
Okay. Okay. And it cannot be done like it has to be between like within a chain second or third carbon. Like these are the first alpha carbon, the second alpha carbon.
Right.
And then the third alpha carbon. I cannot do it like first one.
No. There is no free angle for the first one. On the x axis. True. And if you take this, it's not a stable model, but if you take this and then each time, so let's ignore the first one, but each time put 90 degrees here and 0 degrees here, then 90 degrees. It's not the right model to do it with. Then 90 degrees. So you gradually give yourself a left-handed alpha helix.
Okay. Like this, it will be.
Yeah, an alpha helix in which you'll see that the hydrogen bonds are possible between the first position and the fifth position, or 4 amino acids down or up the polypeptide. The interaction will be between these first and fifth. So you don't have enough here, but
And how do we measure the angles between?
so it's a mixture of a theoretical more simulation, computer simulation, and then there were some experiments that verified the outcome.
So if you inside, normally we don't get alpha helix structure. We get higher order structure like tertiary or quaternary structure. So he broke down to alpha helix and then.
You don't even have, if you have the crystallography structure of a protein, so you can measure whatever you want. Angles, distances, whatever you want. Right? The question is it true ideological structure, but that's a different question. If you have the crystallography structure that you can start feed it into a computer and measure the angles.
And the evolution thing, what does make them to change? Like, is the ancestral change? What is the cause for the change?
That's mechanisms in evolution, which are, in evolution it's mutations. If you each time you have random mutations, and each time the mutation is advantages.
Like one amino acid is getting replaced by something.
Right, by another one. And if the function of the organism becomes better than the basic argument, assumption, I don't know how to call it in evolution, then it will spread this mutation to its offspring, and the mutation will be spread in the population. But it's not really useful. It's just for you to be familiar with the ancestors, the common ancestor term in genetics. אוקיי.
המבחן הקודם.
כן, התחלנו בשיעור האחרון, וזה השיעור האחרון של הקורס, זה כאילו ממש שעשינו את זה ביומן, וזה מותר בגלל שזה לא, כי הקורס בנוי לא רק מהמבחן, זה לא סופי, אלא מבחן עם לעבודה, 70-30. העבודה היא עמוד אחד, 600 מילה, צריך לעלות עם 3 שאלות שאני אתן לכם אותם בהמשך הסמסטר, שצריך לבחור מאמר.
כאילו לכל אחד יש שאלה.
זהו, את בוחרת מאמר שמעניין אותך, שמתעסק בביוכימיה עושה כאילו אנליזה בצורה דומה למאמרים בסמסטר, אז כאילו הסגנון או כמה עמוק וכו' דומה למה שנעשה בכיתה. כאילו יש מאמר שמקשר בין משהו ביוכימי, which means, לא יודע, מולקולה, עד לתופעה נוירולוגית.
אז יש ניתוח של התופעה הנוירולוגית שמוצגת במאמר?
אבל אתה יודע, כאילו זה, השאלות הן די פשוטות, תסבירי מה החלק הביוכימי במאמר, פחות או יותר, או מה הם הוכיחו ביוכימית, מה הם הוכיחו, או על איזה תופעה נוירולוגית מדובר עליו וזה, ועכשיו בעצם קישור בין השניים, כי זה דבר שאנחנו בין קשה לעשות. בגדול זה זה. רגע, את מי שהתכתבת איתי?
כן.
אוקיי.
מה?
את לא...
כמה? מה זה פולקול?
כן, בוא נעלה.
אוקיי.
באיזה תואר?
אני ביולוגיה נקודה קוגניציה. רגע, מה רגע יהיה כל שבוע?
לא, ממש לא. יהיה משהו כמו 4 במהלך הסמסטר, הם גם מופיעים כבר באתר, אבל אני אגיד מתי לקרוא אותם, לא צריך. נגיד שנציין את הפרוט אינסטרקשן פונקשן, אז נקרא... רגע, אז את התכתבתי לגבי לפתוח את המודל או לא? לא. אז את רשומה לקורס? אם את לא יכולה להיכנס לתוך המודל, כנראה שלא. שאז אני לא יודע... פנינה, אני איתך אין לי שום גישה ללרשום, ואם זה לא מסתדר תגידי לי. בכל זאת אני יכול להעביר לך באימייל, את במיילים ליסט?
I have it because in the beginning everyone was
כן, אצלי זה מיילים ליסט, לא, לקורס.
לא, זה לא הייתי קודם.
אז תשלחי לי לכתובת הזאת, זה הכל אין שם, תשלחי לי לפה ואני אצרף לך במיילים. ואותו אני יכול לשלוח לך את המצגות, כאילו, אם לא היה כניסה למודל, אז טוב, תחתבי.
רגע, והעבודה תהיה אחרי הסמסטר?
העבודה תהיה להגיד שבועיים אחרי הסוף נראה לי.
it's still at home and how do you say it?
Actually, I emailed you that I don't want to sign up for
Right, so I think we should ask Ruth. So, are you from the guest school? Which?
Cell and developmental biology, the non-neuro.
Masters?
Yeah.
All right. So you registered to the other courses. How did you do that? Which means what? Go into the
Website
and then put the number and you tried it the same here. And then what? You try to go into the Moodle?
It says it's very ambiguous. It's like either this course is full or you need.
It's not full. There's no limit here. Maybe we can ask Ruthie. She might. This is the secretary down the hall. Let's go together. Maybe she'll be able to. I don't know. Register you manually. So after proteins are synthesized, they undergo several modifications in the cell. Let's introduce these modifications, starting with folding. And starting in vitro. Okay, so in vitro meaning in the test tube. Vitro is the same, comes from the same word as vitrage, the glass decoration found in chapels and so on. So it's in vitro. That's how it's easy to remember. So in vitro means in the test tube, in opposed to in vivo means inside a living organism. So in vitro, and now we're talking hypothetically, any polypeptide chain containing n residues could, in principle, fall into 8 to the power of n conformations. Well, 8 because there's some argument. There is an argument that 8 bond angles are stereochemically allowed. It's not that accurate, and the 8 is not that critical here. The thing is to understand that the point here is that so without any other additional constraints, any polypeptide can attain a lot, also just really a lot of different conformations. But what we find in nature, or even in the test tube, is that all molecules, practically almost all molecules of any protein species, adopt just a single conformation that is called the native state. And for most of these proteins, the native state is the most stably, thermodynamically, the most stably folded form of the molecule. So if we'll plot a hypothetical axis of energy, and here would be conformations. So it should be a multidimensional plane or axis, but these will be conformations of a single protein. So for each one, you'll have a different energy level, and it will be something like this. So for most of the proteins, the native state is right here, with the minimal, absolute minimum in the energy level. All right. And in vivo, and this might take a while before the protein will find itself in the minimum here, because it's a random process, and the different conformations could exist, and you can of course be stuck in a local minimum right here. So that's what happens in vitro. And in vivo, it's a bit different. Folding of proteins is promoted by chaperones. It's the name of a protein family. And there are two kinds of chaperones in the cell. The first, molecular chaperones, and the second, chaperonins. The first kind are small molecules, small proteins that bind and stabilize and mainly avoid, do not allow the protein to undergo improper folding, and thereby preventing aggregation and degradation. And chaperonins, which are bigger molecules, which directly facilitate the folding of proteins. What do we mean by directly facilitate? Let's see it in the next slide. So these are really two distinct proteins. Again, the first one, molecular chaperones, bind the nascent polypeptide. By nascent, we mean as it is being produced, as it is being synthesized on the ribosome and elongated. Already then, the molecular chaperones stick to it, to the polypeptide, oppose any local foldings that do not need to take place. And then for some proteins, it's enough to gain their properly folded form. And for others, the molecular chaperones avoid improper folding and also guide them to the chaperonin, a huge tank-like, tank-shaped protein. The folding protein is inserted into it, and there's also a cap that's being sealed right here. And inside the chaperonin, the protein attains its proper folding. Okay, let's just note that molecular chaperones consist of the HSP, heat shock protein 70, and its homologs. Just an anecdote, but do remember it. There are some nice models, some nice simulations that try to understand better what exactly happened in the chaperones. So I'll show you just the consensus between these different models, again, of what happened, of how the protein attain its proper folding better inside the chaperone than outside of it. So again, let's plot what's supposed to be a multidimensional space, but let's plot just two dimensions in it. These are the different conformations. Let's call it 1 and 2. For a protein, let's say for a protein X, let's say that this will be the initial state, the initial almost linear conformation, which is what happened when the protein is fully synthesized. So this will be the starting point, and this will be the native state when you try to get. And without the chaperoning, the protein has to screen or probe this space until it finds itself to the native state, to the most thermodynamically state. So there should be a z-axis here that describes the energy, right? When I won't plot it. So although the tendency will be to go here, it's a stochastic process, and it might take a while, and it might do something like some trajectory or path like this. And what happens inside the chaperones is that some conformation are simply not allowed. So in a way, this space really decreased in size. All this region of the space is now not allowed inside the chaperones for protein X. And now the path that the protein needs to make is much smaller, right? So it happens faster and more effectively. And you can see papers that deal, with the details of these models, but that's the basics. All right, let's see a short clip from the Molecular Cell Biology by Lodish website, where you can find all these short clips. All right. It's about, to give you some animation about the protein.
In vitro, a denatured protein can refold into its native state on its own. Nonetheless, in vivo protein folding is aided by chaperones in order to increase efficiency and prevent aggregation of misfolded proteins. Molecular chaperones, such as HSP70, assume an open form when bound to ATP. In this form, the chaperones bind nascent polypeptide chains as they're synthesized on ribosomes. The bound ATP is hydrolyzed to ADP. ADP exchange with a new ATP molecule causes the chaperones to switch to a closed conformation, releasing the target protein. A second class of chaperones, the chaperonins, is required to help a small proportion of proteins fold properly. A partially folded or misfolded protein is inserted into the cavity of the chaperonin, where it can fold into its native conformation. ATP binding causes the chaperonin to expand and release the protein properly folded.
So these two kinds of proteins use ATP to do the function to properly fold the protein. Now about chemical modifications. So proteins, after they are synthesized on the ribosome, almost all of them undergo some chemical modification or another. And the most common one is the linkage of a chemical group called, still generally. So the modification is just a linkage of a chemical group to either one of the sides of the ends of the polypeptide, either the N or the C terminals, or somewhere into an internal residue. And these modifications alter the activity, lifespan, cellular location, even dictates whether it will be secreted or not. So it affects every single parameter aspect in the life of a protein. And the most common modification is acetylation, which is the addition of an acetyl group, which is CH3CO, you see it right here, CH3CO, to the N-terminal of protein. So this is the N-terminal residue of a protein. Here is its residual group. The rest of the protein is right here, and the SH3CO is the acetyl group linked right here. And this is an acetylated protein, and it's an essential step because non-acetylated proteins are rapidly degraded inside the cell by intracellular proteases. So proteases are proteins that cleave other proteins and lead to their degradation. And it says here intracellular because there are also extracellular, outside of the cell, proteases. Okay, more modifications other than the acetylation. And these are examples of modification on internal residues of somewhere in the protein. And all of them obviously affect biological processes. So glycosylation, linkage of a saccharide, which we've discussed last time, to a protein. Secreted proteins usually undergo this modification of glycosylation. It's thought that outside the cell, in the more harsh environment, there glycosylation have a protective role. Acetylation can also occur not only in the N-terminal, but also on an internal residue, such as right here, acetyl-lysine. So this is the lysine amino acid, and now it has an acetyl, so it's acetyl-lysine. We've said that there are 20 amino acids, right? So practically, when taking the chemical modifications into account, there are actually much more, because for every amino acid, there are several chemical modifications that can occur. Phosphorylation, the addition of a phosphate. Here it occurs on a serine. Hydroxylation, methylation, the addition of a methyl group. Carboxylation. Okay, each of these modifications is linked to a specific biological process or gives some characteristics to the molecule. You can look it up just a bit, maybe the first three in Wikipedia, just to get a feeling of what these modifications do. What are their effects? Another kind of modifications that is not really the addition of a chemical group is processing. And quite a general term, but this is what we call, in the context of modification, it means removal of specific fragments in the peptide after synthesis. And this process activates enzymes that function in processes such as blood coagulation, programmed cell death, where you have to have a very precise timing. And the idea is that the cell needs to respond for some stimulus, and instead of synthesize the protein from fresh, you take an existing protein, remove some fragment in it, and all of a sudden it becomes functional. Something like a knife inside a sheath. Then, given some stimulus, the sheath is taken out and the knife is active, is functional. So it happens quickly, such as in blood coagulation. So when we are injured, for instance, and start bleeding, some fragments from some proteins are removed, and the cascade of coagulation starts. Because if it not be fast enough, the animal might bleed to death. And so this is common. I think it was discovered first in blood coagulation. Sorry. So this is the processing modification of proteins, when you remove some parts and the protein gain functionality. Okay, degradation. The activity of a protein, first of all, we should state that it depends on the amount of protein present, which reflects the balance between the rate of synthesis and the rate of degradation. I think a decade or maybe 2 decades ago, it was thought that it reflects only the rate of synthesis, and the degradation is a relatively passive process. But now we know there is an active, regulated process of degradation. So we have, you have two sides of the coin. And the lifespan of proteins can be short and long, depends on which tissue you look at. So it can be as short as a few minutes for mitotic cyclins. Again, proteins, a process that has to be very accurate in time, and can be as long as the age of the organism for proteins, for the photoreceptors in the lens of the eye. So you're born with a photoreceptor, and not always, but sometimes you die with the same protein. And degradation of extracellular proteins takes place inside lysosomes. We will see this organelle more into details later on, but these are membrane-limited organelles with acidic interior. So anything that was secreted and now needs to be degraded is taken up again, engulfed into the cytosol through lysosomes, and being degraded. So that's what happens extracellularly. But intracellularly, inside the cytosol, the degradation is by a process called ubiquitination. It's a chemical modification, another kind of chemical modification of a lysine chain, always occurs on a lysine chain, by the addition of a molecule called ubiquitin. Ubiquitin is a 76 residue polypeptide, small protein by itself, but it is linked to other proteins, and it leads to degradation of the ubiquitin tagged protein. So, first of all, this process was revealed, and its particulars, all the details were revealed by these two scientists from the Technion who won the Nobel Prize for it, Nobel and Tchekhanov and Hershko. And let's go over briefly the cascade of ubiquitination. Let's start here before we go back. So this is the target protein, the one that needs to be ubiquitinated and then degraded. So there are two preliminary steps before ubiquitination, meaning loading of a ubiquitin on the protein. First, this E1 ubiquitin activating enzyme has, together with ATP, be ubiquitinated itself, which will then be able to to transfer the ubiquitin to E2, the ubiquitin conjugating enzyme, which will then, together with E3, which is the ubiquitin ligase, enable linkage of the ubiquitin to the lysine chain of this target protein. And these three steps happen not once but several times until the protein is sufficiently ubiquitinated, and then in this state it is tagged, it is recognized to be guided to the proteasome, which is a protein slaughter machine, a huge complex that breaks down proteins back to their building blocks, to peptides, and later on to amino acids. Okay, you don't have to remember, each and every detail here, but just to get the feeling of what we talk about when we're talking about biochemical cascade. So this is a good example. Okay, ubiquitination. Implicated in every single biological process. These are just examples. Every single biological process we find in the cell, and hence the Nobel. Let's skip the movie. It's not that amazing. Okay. To get a better feeling before we'll get into neural system, let's meet this protein family, enzymes, and later on molecular models. So starting with enzymes. Enzymes are proteins that promote chemical alteration of some molecules. The terminology is that ligands are small molecules that are recognized by the protein. And when a protein changes structure of this ligand, so we call the ligand the substrate. And how do they promote the chemical alteration is by decreasing what is called the activation energy. And I'll say a few more words about that. So we previously said that, for instance, energy, an exothermic reaction, when these are the products and these are the reactants, will happen spontaneously, something like that. But it is not 100% true, because it will have what is called an activation energy, some threshold you need to pass before you slide down to the product's energy level. And this peak here is because of there is some intermediate or transiently existing intermediate species of the molecule that's right in the middle, that has the intermediate structure between the reactants and the product that you have to pass. And what the enzymes do is really decrease the height, the threshold that you need to pass. So without an enzyme, it will look like this. With an enzyme, it will look like this, which will obviously occur much more or much faster. All right, how do they decrease this energetic activation energy? By stabilizing the intermediate species. You will see an example in the next slide. But a few more facts about enzymes. So they have immense catalytic power, which means that with them, they increase the rate of production by many folds. It can be a difference of more than 1 million in effectiveness. And they have high degree of specificity, which means that any structural change in the ligand, and it will no longer be recognized as a ligand. And certain amino acid chains of an enzyme form the of definition. And the active site is composed of two regions. First, the binding site, and then the catalytic site. The binding site is obviously where the substrate binds to the protein, and the catalytic site is the site where the ligand is being changed. So again, the active site is composed of the binding site and the catalytic site. Okay, let's see these principles we discussed on a specific example on the enzyme called PKA, protein kinase A. There are several others, B and C. Let's start with protein with PKA. So what this enzyme does is add a phosphate group to certain amino acids, which is exactly phosphorylation, which we discussed. It has two tertiary domains, small domain and large domain. In the large domain, there is a region right here beneath the red carbon trace. Here is the target peptide, part of the protein that's going to be phosphorylated. So underneath it, on the large domain, there is a region that recognizes specifically this peptide and a different peptide. So the specificity of PKA, in opposed to PKB and C, is mainly here, as the region that links the target peptide. In the small domain, we'll find a region called the glycine lead, or cover, that holds the two substrates, or the substrate and the co-substrate, the nucleotide the ATP, together in close proximity. And the nucleotide binding pocket right here, maybe the active site, is this fissure right here between the two domains. Okay, so the specificity is gained through the structure in the large domain, and the catalytic ability, you'll see in a minute how it happens. Right. That's it said. Okay, we will now zoom in into this side, the active side of PKA, and see exactly in the atomic level what happens and how the phosphate is being transferred from ATP to the target peptide. Okay, and I mean, one last sentence before that. So PKA undergoes substrate-induced split. So we said that the tertiary structure undergo continuous fluctuations, right? And before these two substrates are recognized, the small domain is a bit like this. It's farther than the large domain, and when these two substrates take their place, so the glycine lead closes and lock the two substrates together. This is a tertiary structure effect. Okay, and we're now zooming in. We'll see exactly what happens right here in the course. Okay, so first, this is ATP in black. The first two phosphate and the last red one is the third one. That is going to be transferred to the target peptide, here in blue. Only one amino acid from it, but the rest of the peptide is right here. And everything else, all the charges here are of the charges of amino acids that are part of the active site of PKA. Okay, so this is the initial state. The intermediate state is when these two ions interact with the last phosphate, which destabilize this link and sort of make this link possible. And then the end state will be that the phosphate is transferred to the target protein right here. So this is an intermediate state that is responsible for the threshold here, and that is stabilized in the enzyme. Now still, when you reach this peak, of course, you can slide back, not forward towards the product necessarily, but also you can slide back towards the reactant. It's possible, right? So indeed, the intermediate state slides back sometimes into the initial state. But then when that happens, the glycine lead stays long and push you again towards this direction. And only when you reach the end state, the glycine lead, due to conformational changes, the glycine lead opens and the two substrates, now ADP and phosphorylated target protein, dissociate from the active site. So only in the end state is the glycine lead opens and these two break apart from each other, and then the reaction really stops open conformational states when the two domains are farther from one another, which allow the ADP and the phosphorylated target to diffuse from the active site. In enzymes, in order to improve the reaction efficiency, cells have evolved mechanisms for bringing enzymes in a common pathway together, and the outcome, or in other words, assembly on a common scaffold of different enzymes that have or that promote coupled reactions, and the outcome is that, for instance, in the pyruvate dehydrogenase complex of enzymes that we look at right here, one reaction or the products of one reaction are immediately introduced as reactants for the adjacent reaction. And this is gained mainly by spatial proximity between these two enzymes that promotes the effectiveness of the net reaction, which for pyruvate dehydrogenase is right here. So each part of it is attained through a different enzyme, and its function as a complex where the different enzymes are localized in such a way that the products of one reaction are immediately introduced or in the right position to act as reactants in the second reaction. Right, so that's a feature you'll find in the cell for several enzymatic processes. Now let's introduce molecular models. Again, these are the proteins that are responsible for cellular motility. And it's again conformational changes, not surprisingly, in motor proteins that enable movement by coupling with ATP hydrolysis. We will see ATP hydrolysis in the next slide. Myozymes is an important protein family in this respect. They are the ones that are responsible for the most common molecular motors. They have a head, neck, and tail domain, each of them with a distinct function. This is a schematic representation in a black-bone trace, I think, model. Myozymes have have a neck that also interacts with several regulatory polypeptides, and so this will be the tail here. Yeah, and a neck and a tail domain. The head domain has an actin binding site. Actin is another protein that is part of cellular motility, and it serves as the road or the scaffold on which movement is obtained. It has a nucleotide binding site. ATP is the nucleotide that binds right here. And this is the neck with its, again, what it calls regulatory and essential light chains. These are two regulatory chains. Let's leave it like that. So this is the basic structure of myozyme. And now let's see what is called the mechanism of cellular motility for myosin actin-based motility. The schemes here are for muscles, but it's exactly the same when we talk about organelles that move in the cell. For instance, a vesicle packed with neurotransmitters that were packed somewhere in the soma, in the body, in the central region of the cell, on its way to the synapse. These are the same principles. So we're starting right here. This is the thick filament from which the myosin head protrudes, and it binds on its scaffold, which is the actin, which in muscles is called the thin filament. Right here. Okay. Now, before you have any ADP in the system, so the myosin, surprisingly, is not, because you have no ATP, so you would expect to have a loose system, but it's the other way around, because in the lack of ATP, the myosin binds tightly to the actin. And actually, this is the reason for the phenomenon that is called rigor mortis, or stiffness of death. When you die, so the ATP is not produced anymore. So the muscles become stiff precisely because in the lack of ATP, these myosines bind tightly to the actin. And maybe one molecule is not that big deal to dissociate, but a million such molecules, and it's quite hard to oppose this force. now in the presence, now let's go back to life. When in the presence of ATP that binds into its ADP binding site right here, the head dissociates from the filament, the myosin head. Okay, and then we have hydrolysis. The ATP turns into ADP and P, lone phosphate. These two are still there in the binding site, are not dissociated yet. And in parallel to the hydrolysis, we have pivots. The head pivots and binds to a new actin subunit. So earlier it was right here, and now it moves a bit forward. Okay, up till now we didn't have any movement yet, because the thick and thin filaments are in the same position in relation to one another, right? Or the organelle that is linked to the myosin head right here did not do any step yet. And now the step occurs, conjugated to the lone phosphate release from the binding site. In this step, we have, again, the head pivots and moves the filament, which is called the power stroke. Now the thin filament is pushed forward in relation to the thick filament. And dissociation of ADP from the binding site is what completes the cycle and is now ready to bind a new ATP right here. Okay, so again, just to note that, okay, so in neurons it will look a bit different, but it's precisely the same principles in muscles and inside the cell. Yes.
Yes, why doesn't it walk on the other way? What is the reason?
Conformational structural reasons. Conformational change that is allowed right here after dissociation or hydrolysis of the ATP to ADP and P. It's just it has its direction, and when you have all myosines oriented on the thick filament with the same orientation, they will all have the same conformational change, obviously, because they're all the same protein. Then you have movement in a single direction. Now there's also what is called polarity in actin filaments. Let's leave this for another class. Yes.
How does it not, like, it seems like it's a one-way process, so the myosin heads will keep on moving in a single direction. So I guess they have to come back.
Until the concentration of ATP will decrease substantially in the cytosol.
And then they come back to their original?
I do like this, and then it comes back to the original. In the context of so in the context of muscle, there's an opposite antagonist muscle that takes you back.
Okay.
Right. In the context of organelles that traveling in scaffolds from the soma to the synapse and back, so there is polarity in the roads. They have direction, and some roads go towards the synapse while others go back towards the nucleus. And we'll talk about our cell architecture later on, but that's the base. All right, that's the cycle of cellular motility, myosin actin-based cellular motility. Yeah, in this case, it's worthwhile, I think, to see the movie, to give some myosin cortical cycle.
Myosin motility is coupled to ATP hydrolysis. The following mechanism pertains to myosin 2, but all myosins are thought to move the same way. ATP binding opens the cleft, disrupting the actin binding site. This causes myosin to release the actin filament. With ATP hydrolysis, a conformational change rotates the myosin neck and partially closes the cleft, restoring the actin binding site. Myosin rebinding to actin catalyzes phosphate release and activation of the power stroke. The final step of the cycle is ADP release. Movement of myosin along the actin filament continues as the cycle is repeated. In this case, the myosin molecule is tethered, so the resulting action is sliding of the actin filament.
And you can Google myosin actin-based motility for neuronal vesicles or in neurons or something like that. You'll see really cool movies that are based on the crystallography structures of these proteins. So we think that in the tissue it's something that looks like really what you'll see in the movies. Again, just Google it and it's nice. It gives you a bit of intuition what we think happen in tissue when vesicles are transported towards the synapse. set all these things and signal transduction in Europe. Okay. Let's say a few words about common mechanisms for regulating protein function. We've already actually discussed some of the terms here, but just to make it like to discuss it as a bunch of processes together. So okay, so first of all, the catalytic activity of enzymes or the function of any other protein just as well is regulated to meet the needs of the cell. So the cell has a lot of regulatory processes that can increase or decrease the function of the protein. Allosteric mechanism is the most common regulatory process for protein, and it's defined as any change in a protein's tertiary structure or quaternary structure, but anyway, tertiary and up, that is induced by the binding of an activator or an inhibitor by binding of a molecule to any other site than the active site. Okay, so again, the active site, meaning the catalytic and binding site together, anything that binds there changes the function of the protein is not allosteric mechanism. And anything that binds to the protein anywhere else than this active site is allosteric mechanism, and it happens a lot in proteins. And some examples are phosphorylation and the addition of other chemical groups. Okay, let's go one by one. Cooperative binding is a kind of allosteric mechanism in which the binding of a first molecule increases or decreases the binding affinity to the second molecule, and it can also affect the binding affinity to the third and fourth molecule. An example is the calmodulin protein that has 4 sites of calcium binding, and upon binding of the first calcium to calmodulin, the affinity to the second binding increases. Affinity is how well the protein links the molecule. And upon binding to the second calcium, the affinity of the third binding site increases, and so on for the first. The same thing for the first site. So what I've just described is positive cooperativity as an allosteric mechanism. There's also negative cooperativity, which does exactly the opposite. Lowers more and more the affinity of for the ligand, and it changes the function of the protein. Okay, ligand binding can induce allosteric release of catalytic subunits. Again, the processing step can be induced by one or the other allosteric mechanism, and then change it to a state where it is active or it is disactive or it is not active, inactive. So for instance, for GTP, it's a protein that we will become much more familiar later on, but it's in an on state when it has an ATP in its nucleoside binding site, and it can go allosterically and change it, push it towards an off state. Okay. So GDP, calcium, and also phosphorylation, which we mentioned, are maybe the most widely used modulators of protein activity via allosteric mechanisms. We will see lots of examples as we go along. Okay. So again, so just to mention that just to state that several of the modifications we stated here are served as regulatory processes. Now we will discuss this point, I think, next time because it's a big one. But let's just take this note home that before we want to study a protein structure and function in vitro, or really know what it does or how it interacts with other proteins, we have to purify it. That's the initial step before studying. So a lot of methods were developed to purify a protein, and we will introduce most of them in next class. So again, just a reminder, for next time, please answer the question page for biochemistry of the cell, the first chapter, together or alone at home, and we'll do it together in next class.